All right, in this video I'm going to be talking you through meiosis. We're going to have a couple of meiosis videos. You should know from a previous video that meiosis actually has two divisions. We call the first division meiosis 1, and this is going to be the very first video for meiosis 1. We will start with our cell. Let's just say it's in interphase. We start with a cell. It has a regular nucleus with, we'll call it orange, chromatin in there. So we start from interphase. Now, not all cells do meiosis. This is obviously a cell that will become some sort of a sex cell. In males, it will be a sperm. In females, it will be an egg. The cell has already gone through the S phase. So it has already duplicated its DNA, all right? And so we're going to go straight into the first part of meiosis. So this cell, in fact, all of the stages of meiosis have the same names as the stages in mitosis, except because we have two divisions, instead of just prophase and metaphase, etc., we call it prophase 1, metaphase 1, anaphase 1, telophase 1, then on to prophase 2, metaphase 2, etc. So we will start prophase 1. I'm just going to put P and then 1. And a lot of the same things happen in prophase 1 as they did in prophase in mitosis. The nuclear envelope starts to break down. The chromatin condenses into chromosomes. I'm just going to draw chromosomes. And in fact, you might notice that the chromosomes actually come together. So let's maybe list a couple of these things. Membrane breaks down. Chromosomes condense, and they aren't randomly in the nucleus. They actually come together to form what is called a tetrad. Which is the four sister chromatids of two homologous pairs coming together. So they actually pair up sort of in their homologous pairs. Even in prophase 1. And something special happens, which is called crossing over. Notice that these two chromosomes, a portion of their chromatids have crossed over one another during prophase 1. And in fact, this will ultimately lead to exchanging DNA. And in the future, I will draw these chromosomes, if we can just sort of imagine, they will start to look like this. So you'll notice that these actually, they will exchange a portion of DNA, and now that I've used different colors, these, you can see how they've crossed over. Okay, so that's all prophase 1. Now, of course, we also have the centrioles, and the spindle fibers are forming, and they're moving to opposite ends of the pole as well. Okay, we're going to move down here a little bit. Okay, here we are going to start metaphase 1. And in metaphase of mitosis, chromosomes lined up along the equator of the cell. And they will do that here, except for a little differently. They will line up with their homologous pair. And so they line up with their homologous pair. We've exchanged our DNA during crossing over. We have them all attached to spindle fibers that exist coming from centrosomes didn't want it to look like it was attached to the end of the chromosome there. So we line up along the metaphase plate here, but we line up with our homologous pairs. This is different than we had during mitosis. Mitosis, they all lined up single file. Here they are lining up with their pair.